Thank you so much for the very kind introduction and also for the invitation to share our work. I'll just jump right in as Jonathan Omar did. Thank you for the opportunity to speak today in this amazing forum. I like to focus today's sort of sharing, really nicely after Jonathan Omar's work on thinking about what are the toolbox available for us to perform large scale, particularly large sequence engineering. First, I wanted to quickly really thank my whole group. They really did the work, and we are a wonderful kind of family here at Stanford. And my group has three broadly speaking sub-teams. One of the team that I'm going to focus on presenting today is the first part on nature-inspired genome engineering tool development. And second part of my group focus on single-cell approaches for perturbing and tracking cell fate using gene editing systems, really related to my postdoc work with Aviv Regav's group. And finally, we also have a very strong interest connected to Dr. Snyder's talk about how we can leverage AI machine learning approaches, which has emerged, as Dr. Snyder mentioned in his sharing today, really transforming a lot of ways we think about these gene and cell engineering approaches. And today, so I want to really focus more on the first part, connecting to Dr. Jonathan Doudna and a lot of the other speakers on how we think about new tools for better ways, more precise ability to engineer the genome. Early generations of editors, as explained earlier, really are introducing a specific DNA modifications and allows the endogenous cellular repair, showing the bottom of this figure, to really carry out the actual edits. So it's not really doing the whole process of work. And that leads to, for example, a lot of competing events, such as off targets, indels, errors. And as really nicely illustrated earlier by Jonathan's Omar's group, there are a lot of new possibilities now with base editing, prime editing, paste, and a lot of new tools, because now we really are controlling almost the entire editing process by not just having the CRISPR targeting a specific sequence, but also having additional enzymes such as base editing domains, APOBAC, and I'm sure we will hear a lot more also from Billy Lee, another speaker today. And then, for example, reverse transcriptase and integrases. And the limitation here is a lot of times they're great at making short edits, but still a little bit challenging for making, for example, kilobases of edits that a lot of times are really needed for therapeutic payload insertion. So recently, the emerging toolbox for large-scale genome insertion engineering has been really exciting, from the transposes shown on the left, and then the paste and related approaches as Omar and Jonathan shared earlier, as well as additional sort of integrase or recombinase mediated systems. And all the systems listed here, among a few others that are emerging, such as retrotransposons by Dr. Collins' group at Berkeley and others, are really focused on generating, for example, double-stranded DNA, and then using enzymes to integrate double-stranded DNA. And we were interested in thinking about, is there other approaches exist in nature that allows these large genome integration? And what we found that there is a family of recombinases that are called single-strand annealing protein, which actually are mediating single-strand homology insertion in nature. And these are one of the simplest homology repair system in nature, oftentimes most well-known encoded by bacteriophage, which is really probably the simplest organisms, or some people don't even consider them organisms, but they actually possesses a recombination system with only two proteins needed for the entire repair process to happen. So one protein called isonuclease will bind to for example, in this case, a linear DNA, but it can be also other type of DNA, but essentially it binds to double-stranded DNA and revealing a single-stranded ends at both ends of the DNA. And the single-strand region will need to carry homology to a target sequence. And then the core activity is really the single-strand annealing protein shown in green color, sometimes called beta, just because it's kind of alpha beta is actually the second protein kind of described in that phage originally. So that the beta single-strand annealing protein, or we call it SSAP, will mediate a homology single-strand invasion of a donor sequence. And that invasion allows, for example, the yellow part of the sequence, which is an exogenous sequence. Sometimes actually phage would use this system to, for example, insert a whole piece of the phage DNA into the bacterial genome. So in this case, the yellow part will basically be inserted into the blue target DNA through the single-strand annealing process mediated by the beta SSAP protein. These proteins are very compact, usually 200 to 300 amino acid. So it's one of the simplest and smallest systems for DNA repair. Of course, the question becomes, they're encoded by phage, so would they actually work in human? But perhaps not so surprising, thanks to the pioneering work, right, by a lot of the CRISPR, biologists and such as Dr. Zhang. We know that these bacterial systems and microbial systems can be engineered to work in mammalian human cells. So earlier we published work showing that if we use dCas9 to target these SSAP, again shown here in blue, in green, if we can recruit SSAP through a RNA binding protein in blue, MCP, so MS2 RNA binding protein, that essentially have the green SSAP protein be recruited to a dCas9, so that the dCas9 will be programmable through guide RNA to target specific regions of the genome, and the SSAP will hopefully anneal the donor DNA, allow insertion mediated by homology. Then we show that we can potentially insert a large sequence, such as, in this case, a fluorescent tag of about 1 kilobases into the endogenous genome through this homology single-strand annealing. And the system is relatively straightforward if you think about the components, right, on the right. So you have the dCas, basically the CRISPR-Cas9, you have a donor DNA, and then different from the typical Cas9 system, you add a SSAP protein to the process. And we've shown that it can insert in-frame sequence knock-in of, for example, like I mentioned, a fluorescent tag in various loci within the endogenous genome in vitro. This is in cell lines. And we recently have started to expand this work and try to use this for in vivo gene editing. And because we observe that there are, for example, shown here, variable efficiencies of the system depending on the specific locus, I wondered, can we find a more robust system by nature, essentially looking into the natural diversity of these SSAPs? And indeed, because these are phage-encoded proteins, we can find hundreds of them in NCBI sort of phage genomic databases shown on the left. You can see we can reconstruct evolutionary phylogenies based on their evolutionary relationships, and then we can take these evolutionary relationship sample, the natural diversity, and we picked about 600 of these and run a screening to understand if they actually can mediate gene insertion in human cells. And so in the middle is basically activity of these protein at 2 locus we tested. Vertical is one locus, horizontal is another locus. And you can see that there's a really great correlation between locus. And some of the new SSAP we recently screened, this has not been published, but we're hoping to preprint this very soon, is that we found better SSAP versus the Gen 1, which is the published version of the SSAP that we had already tested before. And we validated, clone out these top candidates and individually validated them to have usually 2 to 3-fold efficiency versus the previous published version. And you can see that the activity of these top candidates seem to be really consistent across the 2 different locus we tested. So we now take these to targeting primary cells as a potential way for in vivo therapeutic implementation. So in this case, we're because the size of the SCP is relatively compact, we can package them into AAV with the donor DNA. In this case, we were using a separate delivery of Cas9, but in general, we can essentially use viral delivery, in this case AAV, to deliver the system as well as the donor DNA into primary hepatocytes. And then we show that, for example, the newly identified SSAP definitely performs better versus the published original one. And in collaboration with a with a startup partner, we have started to move this in vivo using more therapeutic relevant delivery systems. In this case, we're combining LNP delivery of mRNA and guide RNA, which is more convenient, more potent, with AAV delivery of the cargo, as shown previously. And we're still running larger experiment, but in collaboration with the startup that are incubating the technology, we're able to show preliminarily that there is definitely efficient sort of insertion in the mouse liver using this approach of combining LNP with AAV. We are still, expanding the work to more animals so that we can reliably quantify the efficiency of the insertion, and we're still doing a lot of genome sequencing experiment to characterize the exact, precision and efficiency in vivo. But we think that with, this potent viral and non-viral delivery approach, we could deliver these SSAP, sort of constructs along with donor DNA into adult animal. In this case, liver, of course, is a better way, is a very convenient place to start. But hopefully, with better engineering of delivery system, like the previous speaker mentioned, and also we will see later today, we hopefully can target a broader scope of sort of tissues and organs using this approach. And finally, I want to share a little bit more about our ongoing thinking about the SSAP landscape. So we actually more recently used a hidden Markov model-based mining approach, which allows examination of less similar sequence if you're just using, for example, simple sequence alignment or BLAST. So this HHMIM mediated search approach combined with a larger database. Now we're expanding our search to gut, microbiome metagenomic databases as well as IMG VR databases. We can now really identify a very large numbers of these SSAPs in nature, over 26,000 candidates that we were able to map in this new search. And we can actually map the activity of our previous characterized, couple hundred SSAP into this tree, so that we can now think about, which part of the evolutionary tree might give us better candidate potentially for human genome engineering. So we're now picking more candidates based on this extended, refined search, and to try to identify even more potent SSAP for in vivo gene editing. So we're very excited about this new expanded search. And of note, I don't have yet a lot of data, but we actually found a lot of SSP are encoded by eukaryotic viruses, even human viruses. So these are going to be proteins that might be naturally evolved to work in human cells. So we're very excited about testing these essentially, kind of human compatible SSAP, hopefully, in gene editing. And very recently, as we think about how to improve SSP editing, we wondered what are the actual three-dimensional structure of this SSP. And as we were looking, luckily there is two labs actually published fairly recently the first ever SSP structure in complex with double-stranded DNA. So you can see how from this structure, the SSP were able to anneal complementary DNA strand, essentially homology sequences. And you can see that they form this very narrow channel that allows two strands of DNA to be inserted and annealed. And what these groups' work showed is that essentially you need very perfect matching of the two strands in order for SSP to form the completed sort of, continuous channel, protein channel. And this really allows us to consider this as a mechanistic understanding of how SAP were able to mediate single-strand invasion into, for example, homologous genome. So we're very excited to now take these structure knowledge and think about if we can, for example, perform mutagenesis, other rational designs to improve SSAP. And of note, we previously did some truncation study even before we learned about the structure work. But you can see that the truncated parts are actually the parts that has been shown in the structure now that are not directly contacting the DNA. So actually the experimental work and the structure work actually aligns really well with each other. So this strengthens or hope that through this rational engineering, we can improve the activity of SAP even further. So in general, we're taking multiple approaches now to try to engineer better versions of SSAP. Hopefully, we can call them next-gen SSAP for in vivo gene editing. We're both sampling a bigger diversity in nature and also taking a more rational approach. And in the meantime, we also are very interested in understanding the mechanism of large DNA repair. And we have some preliminary data showing that some of the pathways, which I don't have time to show today, but we have some preliminary data showing that the mismatch repair, which are important for example, for prime editing, are implicated in these SAP functions. And we hopefully, with novel delivery system from our collaborating teams, we can now really take this system in vivo. So with that, I want to really thank the organizers, and we hope that SAP will provide a additional approach in one of the toolbox for large gene insertion for therapeutic and other applications in the future. Thank you very much. Sorry. I want to thank our founders and the lab for the amazing work. Thank you.